G'day, good morning, good evening. Thank you for listening. This is a medical cannabis show, and I respect the law, and I hope you do too. This information is for scientific and educational purposes. The papers shown will be put in the description. This information and the information's fact-checked, so you can rely on it. And today is no topic. So if you're rewatching this back, you might want to fast forward till you see some slides come across. I show you university slides from my studies related to plant science, soil science, and microbiology. That is within the last 5 years. So you know it's relatively recent and you can rely on it because science is forever changing. But yeah, if you're watching this back, you might want to skip forward to some slides, and that's usually generally the point where it starts at around 5 or 10 minutes. If it only goes for 10 minutes, it means it hasn't been a show today. There's no topic. Excuse me. I rely on the cool folk in chat to ask questions and put discussion topics in for the show to have its title. That's how I come to it now. I used to do it where I go through the latest news and information, that sort of stuff, but it was too much effort for me to do. The little bit that I get out of it, I don't get paid or anything for this, so it's very difficult. It's just a love that I do from my studies and experience. G'day 420 Fred. So it's up to 2 minutes now, and I got to say something. So please jump into. There's only 2 people watching. Let's what can I talk about? The weather is always something forever changing until the show starts. So actually, it's equinox time, isn't it? Yes, it is. The time when the sun crosses the equator. Let's have a look at what the equinox is. Equinox, there you go. Put it into Thingo. So the equinox is a moment when the sun crosses the Earth's equator, when it says to appear directly above it. So is it transitioning in the seasons? Actually, there's a cool chart down here. Check this out. Here's a contour plot of the hours of daylight to correspond with a different latitude. So on the x-axis is the day of non-leap year. So it's the days 10 up to 360. And the y-axis is the latitude from Antarctica to the Tropic of Capricorn, Tropic of Cancer to the Arctic. So it transitions from the right up the north to the south. And if you want to spend time so you can look at where you are, so if you're on the equator, you're just going to get 12 hours. If you're on the Tropic of Cancer, you're going to get 14 hours. So it's right at September there. So this is for the whole year, so it's pretty cool. So it goes through the equinox and the solstices, and then the nice layout. I'll put this in the description later on where you can get it actually. And what it means in plants, it's called PhotoPeriodism. So photoperiodism is the response to changes in day length that enables plants to adapt to seasonal changes in their environment. So you have short day plants. Oh, yeah, here it is, rad. Short day plants and long day plants. Depends what cycle your genus, your plant that you have is in for it to, like brassicas, for instance. They're already done, but other plants, there's good times. Let's anyway get back to it. See if there's, all right, cool. Hang on, put that there. Hello, Lene. Nice to see you. haven't really got a topic for today yet. I was just talking about the equinox and the photoperiodism. How you going, Raptor? So if you want to put in some sort of discussion topic, that would be cool. Otherwise, I'll just get back to photoperiodism because it's pretty much the time. It's very suitable. This is always a reliable website to go through. Photoperiodism. Anything interesting in here? Let's see what the description of these are. Cycle of phytochrome in plants. The phytochrome is the red pigment. And red light, far red light. Okay. So long day plants, when long day plants are long, the lights are short. Short day plants, when the days are short, the nights are long. Then that triggers the flowering response. All right, very good. Oh, that's pretty cool. Long day plants going to flowering. No flowering. So flowering is when the light turns on there. Hang on. Dark. Light interrupts. No. That's confusing to me. Without studying it, it's hard to do it live. Let's see if there's anything else interesting here. Examples of long day plants. Examples of short day plants. Well, short day plants flower as days grow shorter. So there you go. There's a few in there. Animals. Oh wow, they've even got a biological clock when they go into their different seasons. They've got a shed, prepare for winter, I suppose. Prepare for summer. Mammals, what do humans do? Not that this the view has been expressed that humans seasonally is largely because of evolutionary baggage. What? And that is a part of the genome of the population that was advantageous in the past, individuals, but it is disadvantageous under the present day. Right. Okay. Let's get back to it. That'll do, actually. There's someone new. Hello, Travis Kendall. Travis Kendall says, hi, mate. I don't know if it is your field, but do you know much about carts? A little bit. Yeah. I lived in Canada for 5 years. If you come into Travers a few times and just say positive stuff, I'll give you that little wrench thing. So yeah, ask your question. If I don't know, mate, someone in chat usually is quite knowledgeable. Raptor says, I have 3 cuttings outside, been keeping a porch light on when sundown hits and have a solar-powered LED on them at night. They're showing 3 weeks into flower, but I wanted the veg longer. Is it an issue if they go back into veg and then into flower in October? No, not at all. The only thing is it takes about 3 weeks for the transition. So as the plant goes through and shuffles its photoperiodism, it takes a few weeks to transition. So you can expect to see virtually nothing for minimum 2, maximum about 4 weeks. Depends how big it is and how extensive it's gone into that cycle. But that's fine, mate. You can do that. And the problem why it happened is that the LED light that was outside, it wasn't bright enough. So the plant uses moonlight, and if you notice when there's full moon out, just like we've had the supermoon of late, you get shadows on the ground. So there's enough light off the moon to do that. So the plant might think that it's just the full moon. So you have to bump it up a bit more, or put it really, really closer, heaps close. Yeah, actually, I can show you a example of a good photoperiodism. Let's have a look at this. And where is it? Looking for one photo. I don't know where it is. It's down here because it's pretty random. It's beyond. Let's have a look in here. Nope. Houses. Rain. Let's do a cycling. There it is. Found it. Photoperiodism. Here's an example for you, Raptor. See how lights are really close? Quite close. Really cool wire pallets in that road. But yeah, there's an example. What else was there? Here's another thing to do with photoperiodism. The amount of hours. Oh, actually, I'll just stick to the question or the topic. Last 2 years it worked fine, but one of the LED torch lights broke. Oh, spewing. That's no good, mate. That's no good. Yeah. So, Travis, if you're still listening, put your question in, mate, because I do know a bit about carts, and if I don't know your answer, someone in chat will help for sure. And that's about it. What do you think of that, Raptor? Is there any other questions I could, or anybody else? Please chime in. All right, I'll see what else I got for photoperiodism. Things are a bit slow at the moment, question-wise. Actually, I should just get back to the other one. See if there's any in here. Senescence, metabolism. Oh, here's another one. Yeah, I got another couple. So see how the lights are relatively close. It's pretty cool, eh? So all of those lights are, that's just their solar panel on the top of it, and there's light underneath it. Oh, yeah. There you go. So see the size of the solar panel. There's just some lights underneath. That's pretty cool. So I don't have to worry about leads and things like that. Actually, there's another photo here. There you go. Bit of a zoomed in one. I haven't quite started it in here. It's just newly set up. So they might flip because of this poor light. So they're trying to assume that this light is going to cover it for everything. And it might, because see the shadows are pretty big. Actually, hang on, look where the shadow is. It's from behind me. So there's a stronger light behind. That light's probably insufficient. That's just a work light. That's not a light to affect the cycle of the plant photoperiodism. There you go. That was quite related. Is 12 hours, etc. enough? Let's do with the time on and some good studies in that. I'll see if I'll stick just to photoperiodism. Different light treatments. Some examples of major screening pigments. Different light treatments. No, see if this is related. Oh no, this is the different spectrums and intensities of the lights in related to different species, and it gives different responses. And that's the references to the papers that if you read back, if you want to go through it all, you're welcome. It's pretty interesting. It just shows that what blues really do, what reds do in these experiments. So they might suggest different things in literature, but to do it with experiments is pretty cool, because sometimes you get different expressions. But anyway, that's not really related, so I'm not going to talk much about it. 12 hours light spectra. What's this thing? Looks pretty cool. Oh, that's the wavelengths broken down. Spectroradiometer. It's pretty cool, eh? So it's like an action absorber spectra graph, but it's broken down in pigments. It's cool. Green to different types of light, and the wavelengths down here, x-axis, the strengths on the y-axis. But anyway, let's see if there's any questions. Oh yeah, Raptor says, thanks, I figured as much, but I'm okay with the extra 2 weeks because I'm not paying for the sunlight. Yeah. And you have to watch though, if you get the re-veg might show different expressions. It activates the branching BRC gene in the plant, and sometimes you can get 5 nodes out of one node, you might get 5 branches. So be careful with that too. Just watch it when it's in about 2 to 4 weeks. Probably in about a month's time, you might have to prune up the bottoms. Travers Kendall says, I mainly catch the replays. There isn't much information about the carts in general. Would like to know more. Can they be diluted with PG or anything? Not with PG. Someone in chat put in the MCT oil name. That's more food safety. The PG and the VG that they use to make it with, what was happening? The PG burns lighter and the VG burns thicker. But can they be diluted? Well, definitely they can. You just got to put in the carrier agent, more of the carrier agent. Cut a long story short. And I haven't done it for years, but it was the MCT oil. I think it was the MCT 80 or something like that, or MCT 100. It was the MCT oil that was good for vaping and consumption, because some of them, they're not as healthy as they say. And if you're going to get something, make sure it's food grade and don't source it from China. China has no food standards, so they can just say and put out whatever they want. So don't source any food products from there. But yeah, they can be diluted. You'd have to fiddle with it or put a very fine hole so you get a syringe in, but yeah, they can. I hope, did that help, mate? Update me on your question if it didn't help. Well, here we go. We'll get back to Raptors and Sood. PG and VG are both in e-cig vapes, but I don't know if they mix well. MCT oil is and there'll be a newer one out that they use now that's a better carrier agent. I'll put it in for you. Carrier agent for carts. Yeah, I'll share my screen. Trying to help. So then, yeah, I'll do Raptors. In a sec. Oh, look in chat. Lene's updated. Very experienced with carts. Don't smoke MCT oil. It's edible, but not healthy to inhale. See, there's some good stuff. Lene says vegetable glycerine. Really, VG, you reckon? Well, there you go. Someone with way more experience than me in it. So carrier agent for carts. Anyone know what carrier oil is good? Try grey beard, BMOG. Also, don't burn at the highest temperature. Yeah, the higher you go up, it's allowing more hydrocarbons and dangerous pollutants to be vaporized. So the lower the temperature, the better that you can do it at. Here you go. Bio-safe, biodegradable. Hang on. No, stop. Fair income. For, I can't remember what are they called. I can't even remember what the name of them is because I don't want to get on the wrong topic. Carrier carts. No, see, I put a. Here's maybe a suggestion. There's a few suggestions in this one. Let's have a look here. 12 carts. Oh, that's the actual blooming cart itself. Okay, it's not helping. Rosin weight carts. Here you go. What's inside the cart? So, what's inside of it? Terpenes, coconut oil, glycerols, and glycerines. And they include, of the glycerol and glycerines, PEG, polyethylene glycol, that's it, VG and PG. I remember in my studies a lot of PEG coming out. PEG's good for tissue culture too. PG breaks down in the body in about 48 hours. You can also find these thinners in cartridges on the shelves. Okay, contaminants. Additives aren't the only thing you have to worry about when it comes to what's inside your cartridge. Just like with normal, your cartridge could contain pesticides and unwanted materials. In fact, 34 out of 39 cartridges entries were disqualified in one of the detections in one of the latest cups. That was because, actually, was it that one? No, I don't want to comment just in case it's wrong. So find out what's inside your cartridge. Cartridges are convenient, but be cautious. Plenty come with, all right, it's not. So that didn't really help. Here's coconut oil is the MCT, the medium chain triglyceride, which can help make oils thin enough to work on but like it said in chat, they're not safe to inhale. But yeah, your PEG. So, or you can just go down to your local shop and source them, see what they've got, like it said here. So I'll leave that up. I'll put, actually did that help? Let's go back to here. VG can cause lipid pneumonia, etc. Just going by what I know, but never made the own carts, says Lene. You can use pure distillate, I prefer that, or live rosin for the purest vape carts. Okay. So yeah, that would be good. But what about to dilute it? So if you're going to dilute, so if you get some distillate, for instance, what would you do to dilute it? Well, here's a WikiHow thing, actually. Let's have a look at that. Oh, that's right. You wrapped out a question. All right, I'll finish this bit with this WikiHow. Okay. We'll go slow. Oh, hang on. Look at this. Is a massive page. Actually, I'll get rid of that so you can read a bit better. Now it's coming up. Here you go. Method 2. As it scrolls down, probably halfway through it, I think there's 4 methods it selects, and this is on method 2. Method 3, with VG, not PG now. Here's method 4, with alcohol. Alcohol I don't really support because it's got too much extracts afterwards with the vapor. If you can smell it, it's not good. You need to purge alcohol to get rid of that, the hydrocarbons. So there's another option. There you go, gear changer. There you go, mate. So that was the other option. Travers Kendall, can you let us know, mate, if that helps you or if you got any more questions? And see how the people in chat really help as well. Oh, hang on, there's a few other things up here. Oh, for sure, Raptor, yeah. A press is a couple hundred worth the fact of needing 0 additives. Yeah. Travis said, I don't even think my cart tells me what type they are. It doesn't matter. You can just, PG's thinner, VG's thicker. So I'd probably, well, it's self-preference. Oh, wow, some carts are refillable. Fair income, I didn't even know that. Yes, it sure blimmin' is. Oh, here we go, a bit of a, I like the safety stuff. Careful, Raptor says, careful, Travers. A lot of products have fillers in them and they can harm you long term. Yeah, the adulterants. Not very good. People cut it to try and make it go longer, and it's usually not a healthy or safe thing that they cut it with because they just do it for money. So that's why you pay top dollar for really healthy things or make it yourself. Let's go back up to Raptor's question. Some little plants, I think. Here we go. Raptor says, thoughts on seeing a seedling by putting it in straight into flower, then back to veg once sexed. Oh. Well, if I was going to sex it, I'd wait. I'd just leave it in veg. By week 5 to week 6, it'll start showing pre-flowers. And then you'll see them right where the nodes are. So if it's, that's the best way that I suggest. And then you don't get those 2-week or 3-week events where it's changing over in the photoperiodism. So, all right, there's 2 ways. If you did do it that way, you'd grow it up for a few weeks. It's small, then you'd flip it, and then you'd wait another 10 to 12 days, and then you'd see it. And then you'd have to flip it back to put it on to 16 or 18, at least 20 hours, and then you'd wait for another 2 weeks. So it might take 3 to 3.5 weeks to do it that way, to sex the plant. You can also get, you're in the States, so you can get these kits that you send into labs and they'll sex it for you. So the price might have come down for that. It might be 30 or 40 bucks now for them to sex your genes and your cultivar that you're using. But I suggest to leave it to grow to 5 to 6 weeks at least, and then look at the pre-flowers. And if you're really worried of regulars, when you go to flip, make sure there's a lot of ethylene around. And ethylene is promotes femaleness in plants. So it will help get rid of the male tendencies or any stress events that might want to make it wander into herming or showing male characteristics. And how do you get ethylene? They're cut fruit. And 3 major types. There's banana, apples, and pears. These are 3 most volatile fruits that emit ethylene. When you cut them, it emits a lot more. Yeah, like if you get a fruit bag, small fruit bag, 1 or 2 apples inside of that is enough. 1 apple is inside enough for that if you've got seeds or seedlings and things like that for ethylene response. I hope that helps, mate. That's a really cool question. Gear changer says smoke medical at the moment. Outback diesel only 26%, but it does the trick. That's the way. That's the way. Nice. Exactly. Very nice. 26 is pretty good. I find under 20, it doesn't do much for me, or I'll have to have too much of it. And funny enough, that's my barbecue temperature. For the lowest temperature, I'll go is -20. That's just below 0 in Fahrenheit. Yeah, I don't enjoy it when it's below that temperature. When I lived in the Arctic for a few years, but at that temperature, it was all right. No worries. Been talking about photoperiodism and carts, but photoperiodism, I suppose, is happening right now. Very important. What other slides? I'll just have a look through, see if I've got any other ones that I can find for photoperiodism. Let's share the screen. Or put your questions in, please. Travers, if that helps. Give us an update. Or anyone else has any other questions, please put them in. On these untitled slides. That makes it hard. Is there any photosynthesis? Hey, look at this, many lectures just on photosynthesis, respiration, no. Phosphorol. Circadian rhythm, maybe that might be. Oh no, this is to do with the ELF gene, the early flowering gene, which is rad. You know, the plant's got 35,000 genes, and if you can manipulate some of them, you've got a winner. And for one manipulation is the early flowering, the ELF3 gene, which I have managed to manipulate. It's fantastic, and it's easy for marker-assisted breeding techniques, marker-assisted selection, because you're looking at something that's going to flower early or finish early. So it's pretty cool. So it's, is that kind of on the topic? Kind of is. So the plant has circadian rhythm as well. Let's see what else is there. Maybe if this is pretty much it. Sunlight and transitional light. Right? Well, that's already one was showed earlier. And there's experiments about the photoperiod. All right, we'll show them too. So with the photoperiod, if you get the hours on, this is a fantastic experiment. It really, truly is. It taught me a lot. So here's an example of having, so you go into flower mode and leave it on 10 hours. This one was left on 11, 12, 13, 14 hours, still activating the florigen gene, and it's in flower mode. And you can see this one, week 6. That's really poor for week 6, but still, it just gives you an idea. It goes down in size. Isn't that amazing? So at 12, whichever one sort of puts it at, you think, okay, yeah, it's doing, well, that's doing poor, mega poor, but they've probably got a virus in that. Genes isn't giving its genetic potential. The other side's a lot better, which is how it should be at week 6. So you can see 12 to up. Actually, that's poor. That's about, you expect that about day 12, day 15 to look like that. But because the lights were on a lot longer, that's a horrible example. The take-home message from this is 12 works all right, but if you've got any applications to lower it, it can actually tighten them up. It was a great experiment. Flowering under different photoperiods allows you to manipulate the compactness, trim, development time, and the yield. Shorter photoperiods tend to be more compact, high flower trim ratio, mature faster, but decrease in yield a little bit. Inverse results for longer photoperiods. They flower were harvested directly from our grown under that and untrimmed. Okay. So it's just an interesting experiment with the photoperiod. Very cool. Very cool. Cheers. Gear Changer says, oh, what? You had pain in your 2 toes and they amputated them on your right foot because of diabetes. Holy mackerel. What? That's no good, mate. Geez. Any excuse to get on the strong... geez, that's no good. Oh, it says underneath, smoke some sherbet, 30%. Wow. On the couch, yes. You'd be nearly wanting some dry sift. All these carts. Hey, Yield Source, how you going, mate? Hey, do you have any questions, Yield Source? Or statements you've heard, weird things lately that I can elaborate on? I was talking about photoperiodism, but yeah, I've done as much as I can on photoperiodism, I think, without repeating and talking about stuff that's not relevant. So I'm not sure what to talk about. And I'd like to stay on topic unless viewers have got questions. Like this has a question mark. Yield Source says, so overall best flower cycle? Well, it depends. If you're struggling, like it, just pull up a photo. If you're struggling and in an environment that has no CO2 to enrich it, for instance, you might want to drop the hours a little bit. So depends on your cultivar. It might be more happy in 10 or 11 hours of light, not 12. So that'd be 10 on and 14 off. And if you're coming into a hot period, like where it's too hot, for instance, and you want to try and reduce your hours of on time, this might be a good opportunity to try that experiment with your cultivars. But if you're into probably sativa growers, they mightn't like it. Actually, it does say the names. This one was sherbet. Okay. See, they should be nearly, I think this is an Australian one. That's why these results are extremely poor. But look at this, week 8 under 12. That's major problems. Week 8. So yeah. Southern energy, okay. As the two put together. So yeah, really go from that one. Just shows the tightness. So overall best cycle. That's what I'd try. If you've got a lacking in CO2 enriched environment, you might want to drop it down. If you've got a very airy cultivar which doesn't tighten up, which tends to spread its nodes out and stretch, you might want to drop the hours down. So yeah, that's what I'd suggest for the best flower cycle. Green nose, yes. Very odd looking. Yeah, very well. That's Australian, mate. That's what they're used to, I suppose. They don't. I shouldn't say they. I talk a pretty broad reference, but there's some people in Australia that haven't got the education or experience that just don't know what good chunky buds are, how they should be. What's that? 2 fingers wide at day 40, 3 fingers at day 50, and 4 fingers wide at day 60. If you've got no problems in your genetics. And that's tested under many different cultivars. Or when they have problems, it's half that size. Exactly, pretty much half that size. It's just amazing. So it's yeah, day 40, you'd be having 1 finger wide. So put your hand, excuse me, your hands with your tips of your point facing down behind the flowers, and then touch them and you can count how many fingers wide it is. So, yeah, that's what I've found. 2, day 40, 3, 50, 4, day 60. And when there's a problem with a big virus or deficient or some sort of other stress factor, abiotic or biotic stress event involved, major event involved that they didn't get over, it's half that size. So I'd expecting 1 finger at day 40 and 2 fingers at day 60 and be upset, but oh well. Yeah. All right. Now what? Can I finish early? Should I finish early? Or what? Oh, here we go. There's a question. What hormone stimulates tryptophan production? Oh, that's jasmonic acid. Good question, yield source. I'll just prove that to you. Let's get up the pathway for secondary metabolite production. And I want terpenes, trikes it should be. There it is. Let's see if I was right. Jasmonic acid. So what you do, you'll give jasmonic acid in a spray form, or it'll be methylated. It's called methyl, and that's what the plant will recognize. That'll activate the COL1 or the JAS1 gene, which therefore activates the MIB75 or the GL3 gene, which then will initiate trichomes. Depends how it goes into that pathway to if it's going to be a defense or a terpenoid. So, but that's the pathway for them to be activated. There you go. What else? I've got a lot more on. Let's see about some other stuff. Yeah, that's the hormone. There are other ways to activate them. Cloudy. Let's go back into secondary metabolite. I should check, see if there's any more update on the answer to that question. Entourage effect, limonene. Yeah, that's pretty cool. Limonene. Yeah, anyway, too tired. Terpene, very good. Terpene list. No, terpene activation. Right, what's this list thing? And I'll just listed the terpenes, and their therapeutic effect derives from vaporizers at what temperature, its profile, and where you'd expect to find it. This is pretty cool. Yeah, I've been through that for a while. USA company, very nice. Right, back to it. I was going to see if there's any updates. He also says spray it on or feed through medium. I would be spraying it. You're going to get a faster response through that way because it's going to activate it like as if it was getting affected in the field. And that will only take, if you've got prime genes, it can be within minutes. But if you've got unprimed genes, you could expect within hours because it's going to stimulate those pathways like as if a bug or it's received that jasmonic acid from another plant. So through where the other plants allelopathy when they speak to each other. That's the name of that. And I've got some good... All right, let's go. I've got 10 minutes. Let's go into a bit of my plant science stuff and find out exactly should you be doing it. Study. And that information come from my tissue culture, which was plant cell bioprocessing. Oh, what's this? Oh yeah, look at that, it's pretty cool. These are different activations. Photosynthesis happens, then the primary metabolism happens, then it goes to secondary metabolites, then the phenolics, then the different classes of secondary metabolism. Right down, that's cool. What else is in here? Plant's defense. Oh, here we go. Induced plant defense, no. We want, here you go, rad. Yield source, jasmonic acid activates many defense responses through the octadecanoid pathway, and that was an exam question. Jasmonic acid levels rise steeply in response to insect herbivore damage and trigger the production of many proteins involved in plant defenses. Jasmonic acid is synthesized from linolenic acid, which is released from plant membrane lipids. Two organelles participate in jasmonic acid biosynthesis, the chloroplast and the peroxisome. So you want to try and get those jasmonic acid to those two areas, to chloroplasts and peroxisomes, which are in the plant cells. And, you know, the chloroplasts, they're green. So the leaves, so foliar spray would be your answer. Yes. Goes on to say, jasmonic acid is known to induce the transcription of host of genes involved in the defense metabolism, which is called priming the genes. Among these genes it induces are those that encode key enzymes in all the major pathways for secondary metabolite biosynthesis. Several other signaling compounds, including ethylene, salicylic acid, and methyl salicylic, are also induced by insect herbivores. The concerted action of these signaling compounds is necessary for the full activation of induced defenses. Do I have to read all this? Among the diverse components of plant defenses, arsenals induced by jasmonic acid are proteins that interfere with herbivore digestion. For example, some legumes synthesize amylase inhibitors that block the action of the starch digesting enzyme alpha amylase. I don't think I need to go and read all that because it goes a bit more into detail. And we don't have to talk about that because that's, but yeah, that's it. Right, I love thoroughly answering. So I don't have to go through all my slides now to find that slide. Actually, I'm happy with that. Are you happy? Are you happy? Yield source? Because I think that pretty much, that covers it. I can elaborate a lot more in detail if you'd like. Yield source says, nice. So jasmonic acid or synthetically linolenic acid inhibits plant defense and starts that cascade signaling pathway, which is what you want, because you want to activate those, the COL1 and the MIB genes, and those which in turn start producing the defense responses, which is the trichome production. So yes, nothing like the slides, because I might forget, you know, I might be like, that's FL22 receptor that's in the plant that can have responses from bacteria with flagella. You know, the flagella out the back of the little tail thing. that's something I used to get mixed up a lot. So the slides, just keep it, keep it all square and on point and fact checked, because I like to be, it's got to be all thorough. I don't like saying stuff that I even half guess or 3/4 guess. No, because there's no point in putting those facts out. Oh, good. Here we go. Yield source. Yep, I'm happy for sure. Good on you, mate. Good stuff. And you can get, don't go with synthetically linoleic acid. Go with methyl jasmonic acid. Look up for that. You can get it. And depends on your quantity that you need. You'll probably get a little tiny amount. So you're only going to need 1 to 5 grams at the mega most, because you're going to be diluting it such at a low ratio. It'll be parts per million that it'll be activated at. So it might be 1 or 2 parts per million that is sufficient enough to be sprayed on the plant for it to stimulate that pathway. Awesome. Awesome summary. Well, put your questions in or statements in now, because I'm happy with today's show. I can, what can I write it up as? Well, it'll be the last bit, jasmonic acid. What do we do at the start? We're into photoperiodism and carts. So yeah, the 3 things we went through today. Let's hope that YouTube monetizes this, because I think there was no problems at all. Everything was fact checked and everything was good. So I do try and stay on point for educational and scientific purposes, so that this information can be relied on. I'll put some of the links in down below for the description of the photoperiodism and anything else I discussed that's in the paper form. I don't think there's anything else because there's nothing open up the top. But yeah, it's about it. I'll be doing this again next week. I love going through the secondary metabolites. We haven't talked about secondary metabolites for a while. So it's a key thing. Secondary metabolites is everything. Even in vegetables, it's everything, because it's the nutritional value or medicinal value that's high, that's for which we have plants. We don't want to grow things that aren't potent or very, we want to bring out the genetic potential in them. So we want things to be very active, full of vitamins, minerals, and other good secondary metabolites that we need. So yeah, the secondary metabolite pathway is really good to know. Righto. Well, that's about it. Thanks everyone for listening. I'll be here again next week. Bring your questions and statements for them. I appreciate your ears. Happy breeding, happy growing, good health to you all. Bye-bye. Thank you. Bye.